Yeah, sorry I missed everybody today at noon. I had to go into town to get my truck that had died on me earlier. Yeah, that was a now, which is good because we haul our water and our trash out here. We need four-wheel drive when it rains on the weekend. So this week, we've got test number 3 is now up and available to you guys. It's very similar to a test that Dr. Hopkin gave earlier in the semester. It consists of there are 10 short answer / essay questions, accounting for a total of 30 points, and then the rest is some fill in the blank with some multiple choice questions. So first off, let me just kind of remind you, it's over chapters 8, 9, and 10. You can get a lot of the material from, or basically everything you need to know to answer the questions in this test come from the reading in the book and the PowerPoints, okay? Now, I've also got some podcasts out there. The podcast is just another way to deliver this information. And the lecture videos, once again, that's another way for me to deliver the information to you. And however, it works best for you, some of that will work. You can use a mixture of all of those. But like I said, I know that watching YouTube videos, what are we doing now, right? Watching a YouTube video can get tedious, especially trying to look through 30 minutes of a video to find an answer. You like I said, you can return to the book or to the PowerPoints to do that. Oh, all right. Somebody's hitting me up saying hello. So as we go through this, you guys are welcome to shoot questions to me over here in the live chat. I will address your questions as they come in. But before I get started, I know that one thing that I really want to hit home here on this test is question number 1. That is the essay question. I believe it's worth 6 points, which out of a 50-point test, that's 12% of your test. And to me, it's an all-encompassing question. And it asks, compare and contrast oxidative phosphorylation to photophosphorylation. So the idea of compare and contrast, how are they similar? How are they different? And I've instructed the TAs, details matter. Now, you don't have to have an introduction and a conclusion, but what I do want you to tell me is, how are they similar? They're both making ATP with ATP synthase, right? They're both using electron transport chains to create proton gradients, right? Okay, they're both relying on proton motive forces and chemiosmosis. I'm throwing out a lot of vocabulary words. You want to do well on that? You better get those words down. That's what they share in common. Things that are different between photophosphorylation and oxidative phosphorylation. My dog down there is whining. He wants my attention. But basically, the name says it, oxidative versus photophosphorylation. Phosphorylation means we're going to phosphorylate something. Specifically, we're going to add a phosphate group, ADP, to make ATP. And then the oxidative versus photophosphorylation, the big differences are where are the electrons coming from? Because the electrons power the electron transport chain that creates your proton gradient, right? Or your electrochemical gradient. You can use either one of those terminology. The electrons in oxidative phosphorylation are coming directly from NADH and FADH2 in oxidative phosphorylation, and those are actually coming from high energy electrons that you're ripping off of organic molecules. That's also your source of energy to create your proton pump. To create your proton gradient as well. Versus photophosphorylation, the electrons are coming from water. And if you're ripping electrons away from water, just remember, oxygen holds onto those electrons very tightly. Those electrons don't have much energy, so the energy to elevate them is, of course, coming from sunlight. And that is a very key difference between photophosphorylation and oxidative phosphorylation. And another difference is the fate of those electrons. So the electrons matter in these two processes, right? And in oxidative phosphorylation, the electrons, of course, are ending up attached to oxygen, right? That's complex 4, cytochrome complex, which is the protein complex 4, adds those electrons onto oxygen, and then the oxygen picks up the protons, creates water. And in photophosphorylation, electrons flow from water through the photosystems, through the electron transport chain, and end up on NADPH, which is the high energy electron carrier used in photosynthesis. And that carries it, of course, to the CAM cycle where it will be used to reduce carbon into an organic molecule. So the fate of the electrons in these two systems is also quite different. So as you write that question, as you answer that question, make sure that you answer it with details, and be specific on it, because details matter. We're not trying to write a short answer on that one. And which is the last essay question, and it says, what is the most important or interesting thing you learned in this portion of the class? Okay, this is slightly different from Dr. Hopkins. He said in his test, it had to be something that was not addressed in the test. In this case, it can be in the test, but you want to make sure you explain your topic and don't repeat don't repeat one of your other answers. So that is important. Just make sure you don't, if you say you like photophosphorylation is your favorite thing, that's fantastic. You're welcome to talk about it, but make sure that you add details in addition to what you wrote in question number 1. Okay, so the idea is, don't just say see question number 1 or see question number 7 or something. That won't work. You write me a paragraph. Okay, so I'm getting a couple questions. All right, thank you for answering my question. Yep, you're welcome. Is there still an essay question? Of course, that's what I spent the last 5 minutes basically explaining about these essay questions. One other one, number 8. Why is chlorophyll green? All right, there's a simple answer that's correct. Not going to get you full credit by any stretch of the imagination. And then there's a complete answer. The simple answer is because it reflects green light. Yeah, sure, that's right. Why does it reflect green light? That's the question. That's the answer, is to expand on why plants reflect green light, because green light is energy than red light. So why in the world would a plant absorb a light that has less energy and reflect one that has more energy, like the green light compared to red light? So there's a reason for that, and make sure you explain that reason well, and that would be question number 8. Okay. Also, I don't have any other questions right now. Hold on. How long should our response be for the essay question? Well, that's up to you. Do you want to make an A, a B, or a C? The more you write, the more complete your answer, the more credit you're going to get. Okay, question number 9. Explain why a molecule of ATP stores more chemical energy than a molecule of water. Okay, so ATP is the energy currency of life, right? Adenosine triphosphate. We know that molecule carries 7.3 kilocalories a mole of energy. A water molecule has basically very, very, very little energy available in it. And in fact, you know this because it's a very stable molecule. You take a match around water, throw it in the water, the most brutal thing of all happens. Nothing. The match goes out. That's because water has very little chemical energy. And the way you're going to answer that question is you need to go back to chapter 8, and you need to study, look at the slides on potential energy, and specifically chemical energy as it relates to the electrons and their position relative to the nucleus, and read up on ATP and why having a third phosphate group has more potential energy than having just 2 phosphate groups, and how that might relate to how oxygen is holding onto electrons and water. And that would be how you would answer that. Okay, how do can't speak here. How do carbohydrates have more energy stored, but something phosphorylated has more potential energy? Okay, writer, man, love that name, by the way. Okay, I think I'll double check my question and make sure I don't have an extra more in there. We know that carbohydrates store potential energy, right? I mean, we eat carbs for energy. I mean, you have simple carbs give you energy, complex carbs give you a long-term source of energy. You can burn wood, which has cellulose, which is a polymer of glucose molecules, has lots of energy because, we burn wood for fire, right? Okay, so carbohydrates store potential energy. Now, if I take something like a glucose molecule and I phosphorylate it, it has more potential energy. In fact, a lot of times whenever you add a phosphate group to something, you increase that potential energy. And in fact, that's why ATP is often coupled with these endergonic reactions, because the release of energy going from ATP to ADP provides energy to supply an endergonic reactions, that energy coupling, right? And we do that by creating this activated intermediate. So what I would do to answer that question, think about a phosphate group. It's a phosphorus. It's surrounded by oxygens. Oxygens are electronegative. In fact, some of them even lose their protons and become negatively charged. And then you look at a carbohydrate, C6H12O6. What are those oxygens doing? Most of them are forming hydroxyl groups on the carbohydrates, right? Those are polar covalent bonds. So if you have a polar covalent bond, oxygen is going to pick up a slight negative charge. I'm adding on a phosphate group surrounded by oxygens. All those oxygens are negatively charged. Like charges do what? They repel. And when you start sticking phosphate groups on something, all those negative charges start repelling, and that makes way more potential energy. Can you guys still hear me? Let me know if you can hear me. Somebody shoot me a hands up or something. My computer is telling me my internet is bad here. So going back to when you phosphorylate something, all those oxygens start. Thank you guys. Thank you so much. Awesome. All right. I'm still on. Okay. So here we go. My dog's whining. I'm sorry. He just wants my attention. Phosphate groups, lots of negative charges. Stick them on a molecule that has hydroxyl groups, carbonyl groups, carboxyl groups. Remember, all those are C bonded to O's. They have partial negative charges. Those charges repel each other. I don't have a rubber band with me, but you ever taken a rubber band and you stretched it? It has potential energy. It's got elastic energy at that point. It's very similar to a chemical bond where all the things are repelling each other. That's how it stores extra chemical energy. That's why phosphorylating stuff adds energy. So I hope that answers your question, Midnight Rider. Okay. Does photosynthesis reduce entropy since it's creating a stable? That's a fantastic question, Eduardo. I mean, I love those kinds of questions, and that's a good one. And let me see if I can address this. First of all, life never violates the laws of thermodynamics. The first law, you can't create nor destroy energy. So we got a set amount of energy in the universe. This is, of course, a closed system. Nothing gets in, nothing gets out. You can transfer, you can transform energy. I can kick a ball. The motion of my foot moves a ball. That's a transferring of kinetic energy. You can transform energy. You can convert and you can transform it from kinetic energy to potential energy back to kinetic energy. Okay, now life, we don't violate the laws of thermodynamics. We are an open system. Energy flows through life. It's like the force flowing through a Jedi, right? But energy flows through life. So we are an open system. We have a constant supply of energy. Now let's take a look at photosynthesis. Let me make sure here. Does photosynthesis reduce entropy? And the answer is yes and no. All right, I know this is complicated. At the level of the cell, and in fact, all life at the level of the cell creates order through endergonic reactions and maintaining homeostasis. So life is reducing entropy at the level of the cell or the organism in general, because you and I are multicellular organisms made up of trillions of cells, right? So I am an island of low entropy. Let me turn my heater off here. I'm an island of low entropy, and I'm using energy to create that entropy, to lower the entropy, create order, and maintain homeostasis. But I'm emitting energy constantly, like 100 watts, I'm like a 100-watt light bulb. You are too, right? Emitting energy. So we're creating entropy all around us. Let's go to photosynthesis. You're taking carbon dioxide and water, two fairly simple molecules, and you're using energy to create glucose. And yes, glucose has lower entropy than the reactants. So in this case, it's an endergonic reaction. So when you look at like Gibbs free energy, right, that change, we're going to increase the free energy of the system. I'm sorry, we're gonna lower the entropy of the product glucose, right? It's got like 24 atoms in it. So yes, it has lowered the entropy of that reaction. I hope that answers your question right here. And of course, life does not violate the laws of thermodynamics because we are increasing entropy around us, right? So just by existing, you could actually call life an entropy generator in the entire universe but not at the boundary of our bodies or our cells. So we are a system, and we're all these interconnected parts. A cell is all these interconnected parts, and the boundary of the cell as a system is, of course, the cell membrane. For me, and animals and plants, our boundary is like our skin. Okay. In your first email regarding, we only have 40 hours from the moment. Oh, guys, okay. So, yeah, it's just due on Sunday night at midnight. The test is due at 11:59 p.m. Sunday at midnight. So, this was on me. The previous test came due on Thursday. I was going to have mine due on Tuesday. I teach another class where I basically, the test is available on Monday and open all the way until Sunday night. I kind of got my two classes a little confused, and I figured you guys wouldn't mind having a little extra time to do that. And honestly, I thought about it long and hard of changing it. And this week is a rough week for some of us. There's a lot of stress, a lot of tension, and I think we need the extra time on the test this week. Yeah, we need extra time. This is a rough week. It's very tense for a lot of people. Due Sunday midnight. Doesn't matter. 11:59:00. You can open it, spend your time on it. Take your time. Take your time. Use your book. Use your notes. Use Google, Wikipedia. Love Wikipedia. Love Khan Academy, Crash Course. Although that question on endosymbiosis, better be careful because there's a lot of wrong answers out there. And that's like my own personal crusade is to explain why the common view of a proto-eukaryotic cell engulfing an aerobically respiring bacteria is likely wrong. Yeah, that's a test question. Now you guys know it. Be sure you review my material because that is remarkably, I can't believe how often it's wrong and wrongly taught. And we can disprove that proto-eukaryote engulfing a bacteria based on just simple cell biology that we've already learned this semester. Okay. Video is spotty. Good. This is answering question 30 about glucose. Let's see here. All right. I am confused on question number 5. Hey, that's a really good question. Okay. So I'm confused about question number 5. Number 5 is, what do you think were the 2 most important consequences of photosynthesis for life on Earth? Explain why. So I've got a couple videos out there and PowerPoints, and this is not in the book, okay? And basically, it's an evolutionary point of view, and it says, why do we care about photosynthesis? Why does this matter? And basically, photosynthesis transformed the Earth in so many ways and paved the way for multicellular life. And this is something that I really enjoy talking about because, oh, buddy, my dog's just down here whimpering because I'm not paying enough attention to him. He doesn't know what's going on. Okay, so, all right, so we know that bacteria around 3 billion years ago evolved photosynthetic pathways, and especially called oxygenic photosynthesis. There are, believe it or not, there are photosynthetic organisms that don't produce oxygen. They produce elemental sulfur. There's a sulfur bacteria. They use hydrogen sulfide, H2S, as their electron acceptor. As their electron donor, not water. But oxygenic photosynthesis, green things that release oxygen. So here's why it's important, guys. This is amazing. Think about it. Okay, so water is pretty abundant, right? Sunlight's pretty abundant. Early in the Earth's history, carbon dioxide was abundant. And by using energy and sunlight, the carbon dioxide and water, it created carbohydrates and released oxygen. This did 2 things right there straight up. One, by creating glucose, it added a lot more energy and carbon available to the Earth's ecosystems. So by having way more carbon and energy available, the ecosystem's life could really take over. Grow everywhere, especially the oceans anyways, because life didn't actually come on land until the last 5 or so 100 million years ago. But, for the other 3 billion years, life could really live in water because there's a lot more energy and nutrients available to life just by having photosynthesis. Secondly, released oxygen into the atmosphere. This had some profound effects. Oxygen, you know what I'm going to say here. Oxygen is the second most electronegative element in the universe. It loves electrons. It oxidizes everything. So at first, oxygen was damaging to life, but the reality is Oxygen energized life, paving the way for multicellular life. So in response to higher levels of oxygen, aerobic respiration evolved. That's the ability to make a lot more ATP for every molecule of glucose. That led to endosymbiosis, which led to, you can see my puppy in the background. Endosymbiosis led, of course, to the evolution of eukaryotic cells. Eukaryotic cells went on to go form all multicellular life on this planet. There are no multicellular prokaryotes. Think about that. There are no multicellular prokaryotes. All plants, animals, fungus. That's it. Lost millions of species. They're quite diverse. They've taken over the world. Are all made up of eukaryotic cells. So oxygen energized life led to aerobic respiration, led to the endosymbiosis, which led to the evolution of eukaryotic cells, which then led to the evolution of multicellular life, including animal life. So that's important. Now, oxygen and photosynthesis did other things for us besides just paving the way for multicellular life. It also added oxygen to the atmosphere, so it cleaned out all the methane and other volatile organics in the atmosphere. So you like this nice blue skies? That's actually Rayleigh scattering from nitrogen gas. But it also, it's not as hazy. It oxidized all the iron out of the oceans. That's why they're nice and blue today. But it also created an ozone layer. And you guys know that the ozone layer filters out ultraviolet light. So without the ozone layer, we wouldn't have forest on land. There would be no land animals, no forest. And believe it or not, ultraviolet light can actually split water. So over hundreds of millions of years, without an ozone layer, the sunlight would eventually split the water in the Earth's oceans into hydrogen gas and oxygen. The hydrogen gas could not stay on Earth for very long. We're not big enough to hold on to it, so it'd escape into space. Oxygen would react with minerals on the surface, and over time, the Earth would end up looking like Mars, an oxidized, dead, red planet if it weren't for our ozone layer. So I hope that answers why photosynthesis is important. And you don't have to write all of that for your test. You could just pick some of those and make sure that you answer it well. Okay. Can you explain the consequences of photosynthesis? I just explained that one. The extra time. A truly slow test taker. Hey, no worry on the slow test taking. And in fact, I want you guys to seriously take your time on this, dive into this material, look stuff up. I mean, this is your opportunity to go learn this material, sit down, know exactly what we're asking you, and not try to guess what we're going to ask you, but know exactly what was on the test. Have some fun learning the material along the way. And I hope that by having this test available, that it really takes some of that pressure off of you to wonder what we're going to ask, and if you studied it well enough. Now you've got the time to go do it. So you're welcome. The slow test taker, and in fact for test 4, I'll do the same thing. I'll give you guys a few extra days to take it, because I think we need more time these days to do stuff, especially to take these tests. Okay, can I explain a little more endosymbiosis? What we talked about it, what it means. We talked about how could. Okay, sorry, I'm trying to read this really quickly. Endosymbiosis, and what you're asking is, could we go into a bit how it could not have happened? Okay, so I figured somebody would want to know this. I've got several videos on this, guys. In fact, this is one of my big pet peeves in the way things are taught wrongly in biology. So let's just think about endosymbiosis, and endo means within, symbiosis means living together, and this is how we had the evolution of eukaryotic cells. Basically, we're a chimera. Isn't that weird? We're a chimera. What are we? We are this mixture of we're two prokaryotic cells that came together, an archaean, right? And this little aerobically respiring bacteria that probably bored its way right into the archaean. Now, when you go to the web, you type in endosymbiosis, you're going to see some proto-eukaryotic cell with a nucleus and some organelles and some other invaginations engulfing this aerobically respiring bacteria. Well, okay, let's think about that for a second. Okay, how do prokaryotes make energy? They can do it through substrate level phosphorylation. Yeah, good luck making a lot of ATP there, right? But all cells on this planet make ATP using chemiosmosis. Every single cell on this planet uses an electron transport chain to create an electrochemical gradient that is used to power chemiosmosis, right? Making ATP with that ATP synthase. Prokaryotes just do it across their cell membrane. So if a prokaryotic cell gets larger and larger and larger, its volume gets larger at the third power, length, width, height, right? But its surface gets bigger at the second power, length, and width. So as the cell gets bigger and bigger and bigger, whoa, there's a fly on there. Ah, man, it's like Flamageddon out here. So as the cell gets larger and larger and larger, it makes less ATP proportional to its size. That's why prokaryotes are limited in size. It has nothing to do with diffusion across the membrane. In fact, a molecule could diffuse across the entire width of even a eukaryotic cell within milliseconds. And in fact, well, not you, but people can get, and puppies can get worms, like tapeworms. Tapeworms, they can be 20 feet long and body thick. Diffusion works just fine for them, for all their nutrients. So diffusion does not limit the size of a prokaryote. Energy production does. So endosymbiosis is showing the cell getting larger and larger and larger. Well, I mean, that would be really hard if it starts running out of energy. Secondly, what does it take to do phagocytosis, to engulf another cell? Okay, well, you've got to take your cell membrane and wrap it around another cell. Hey, buddy, it's okay. And to do that, you use a cytoskeleton, specifically actin filaments in microtubules, and the motor proteins involved with these things. Okay, in all of our history, we have never discovered a prokaryotic cell capable of phagocytosis because it requires a lot of energy and a complex cytoskeleton to do that. So why would we expect something 2 billion years ago to be able to do phagocytosis? So we have no reason to believe that there was a phagocytotic event causing that, okay? And also, as I just said, the prokaryotic cells are limited in size due to energy production. Buddy, that's your stick. Here you go. He wanted his stick. This is Titus. Hi, Titus. And sorry for this is what happens when you work at home. You get photobombed. There's that fly again. Okay, so that's how we know that endosymbiosis, at least primary endosymbiosis, leading to the evolution of eukaryotic cells, did not occur through a proto-eukaryotic cell causing or phagocytizing. What most likely happened is that the aerobically respiring bacteria was an intracellular parasite. It bored its way into the larger archaean, the other prokaryotic cell, and formed a symbiotic relationship from there. And that is something we do see happen. We do know of parasites, bacterial parasites, that live inside, other cells, including other prokaryotes. So I hope that explains it. And also, one other thing, when it comes to plants, photosynthesis, that's secondary endosymbiosis. And yes, a eukaryotic cell did phagocytize a cyanobacteria about 1.5 billion years ago that led to the evolution of plants, because plants are eukaryotic cells. They have a cell nucleus. They have mitochondria. In effect, right now at night, all of our plants are respiring. They're using oxygen and releasing carbon dioxide into the atmosphere. Okay, hope that helps out. Is quantum effects asking us to why it was meaningful to learn about the potential root cause of some of these biological principles, less about the specifics of the mechanism? You're absolutely right. You guys do not need to explain quantum effects for photosynthesis. Hey, knock it off. Sorry, he's in there trying to find some food. Oh, buddy. Okay, so no, you do not have to explain superposition or quantum tunneling for how enzymes work, or coherence for photosynthesis. You know, that's well beyond the scope of this class. Hey, come here. Let's go out. Come on. How about, buddy? All right. He just needs lots of attention. Okay, so going back to the quantum effects, no, you in no way need to explain how the quantum world works for biology. That is well far beyond this class. And in fact, I'm still trying to wrap my head around how some of this works. But the point that I was trying to make with that is that, you know, in these intro textbooks, we often teach stuff and this is exactly how it works, and we know how it works, and we're just relaying that information to you guys. And the point is that we may not really know. I mean, that's why I have in that slide, I've got like, what's the problem here? Well, when you think about how when you think of the efficiency of photosynthesis, it's like 98% efficient. It is incredibly efficient. So when a photon hits the antenna complex, like 98% of all the energy makes it to the reaction complex. That's unheard of. And people are like, a random walk, that just doesn't work. That goes up in energy, elevates to the next energy, and it just flows downhill to the reaction center because you lose energy at each step, right? And people have questioned that inductive resonance, and said, that random walk just doesn't, the math just doesn't add up. Is there another explanation for this? And so the idea with that is we don't know everything in these, even though we teach it like this is how it works. We don't know everything, and by questioning some of these basic things, we push the science forward, and we may be wrong. I mean, it may not be a quantum effect. It might be just a typical classical physics effect. We don't know yet. But if it is a quantum effect, it could revolutionize the way we think about biological systems in relation to physics. And that's not just theoretical applications, guys. That could be how we build quantum computers. My computer, I mean, I built it myself. It cost me $2,500, but I'm running like an i9 12-core processor, right? It's fast. It's the fastest thing I could just about build on my budget. But a quantum computer would smoke anything we have. So, if we understand how plants are solving this with using quantum computing, amazing. Also, our solar panels, we need energy. Solar cells are getting cheaper and cheaper and cheaper. But if we could figure out how plants are so efficient at taking energy from a photon and translating that to chemical energy, right? Because remember, once you get that energy of the photon to the reaction center and it shoots that electron up and gets picked up by phaophyton, you have now transformed the kinetic energy and electromagnetic radiation into chemical energy. And if we can figure out how to do that, that's awesome. We could greatly improve the efficiency of our solar panels. And also that photosystem too. It's stripping electrons away from oxygen and releasing hydrogen gas, hydrogen fuel cells, hydrogen gas, then we could, like I said, have a lot more energy production. So yeah, studying this and understanding it at these minute quantum details is incredibly important. Okay, I hope that explains it. Yeah, so go to the slides when I talked about it, and I said, this is the problem, this is the question. Hopefully that explains it. Like I said, please don't try to explain quantum coherence and photosynthesis or quantum tunneling and enzymes. I mean, if you can, great. But the answer to that question is, why did I think that was important? And I hope you guys kind of got that right now. Okay. Let's see here. Oh, good. I'm glad you guys asked about that. Number 12. Let's see here. Somebody else said hi, Ty. That put him out. He was going crazy. A system of equilibrium. Okay. Think about this from a qualitative point of view. Equilibrium. If you're in equilibrium, the two states are the same. The two systems are the same. So imagine I've got 2 glasses of water, and they're both right at room temperature. They would be in equilibrium with each other. They'd be in equilibrium with the room. Imagine I've got a glass right here. This is my good old Florida State mug. It's empty. I was drinking some soda earlier. And let's say I put a barrier in here, and it's permeable to water, but not to a solute. And I put some solutes right in this side, and this stays pure water. What will happen is, as you know, through osmosis, the system will now be out of equilibrium, and the water will push its way up into here. Okay, now think about that. These two systems are out of equilibrium. I filled it up and put salt here. Water flows that way. It's flowing in one direction. That's low entropy. Water flowing in one direction, wind flowing in one direction. We can harness that to do work. So delta G, which is your free energy, is less than 0 because you've got energy to do work. It's like water flowing down a hill. River in the Rio Grande can be used to do work because it's slowly flowing downhill because it's not in equilibrium with the ocean or higher up. So that water has kinetic energy. Go up to a lake. The water in cochineal by itself is all in equilibrium with itself. It can't do any work. All right. So basically, if you have, if you're in equilibrium, what's your delta G going to be? If you have energy to do work, your delta G is less than 0. If you have an endergonic reaction where you're requiring energy, your delta G is greater than 0. And if you're in equilibrium, that's as good as I'm going to give you. Okay. Do we need to submit the essay answer separately? No, put everything in Learn. Like, so what I would suggest doing is typing it up in a Word document, because Word document, it flags, oh my gosh, it tells you everything you're doing wrong. Write it, correct it, write it well, edit it a couple of times, and then cut and paste your answers directly into Learn. But I suggest typing out your answers, write them all in one over a couple of days, look at them over tomorrow and Saturday and Sunday, and then submit, your answers after you've looked at them for a day. Don't wait till the last minute on Sunday night to do it. Okay. Oh, is this one? I absolutely love all the little signs of physics and biology. Not truly blue. Yeah. It's interesting. Okay. I love physics and chemistry. And was it chemistry called themselves the central science, and physics is like, well, the entire universe is based on physics. Like, you're absolutely right. Biology, we're the apex science, because we're dependent on both physics, both classical physics, which is like, think Newton, like ball and stick models, force of gravity, objects in motion stay in motion, that kind of stuff. And we depend on quantum effects as well, like the weird world of atoms and how they move in waves and particles, and they can be entangled with each other and weird. We depend on that too. And of course, we depend on chemistry. We are the apex science. And in fact, climate science, geology, we're dependent on astronomy. Yes, they encompass all the sciences. That's what makes biology so cool. Glad you like it. Okay. If how mitochondria produce ATP, but I'm having trouble understanding how to produce ATP without mitochondria. Crystal, that's yeah. So there are two different ways that cells make ATP. The first one is substrate level phosphorylation. Think glycolysis. Think Krebs cycle. Prokaryotes do that too. And the way they do that is they're going to take the breakdown of an organic molecule like glucose or a lipid, and that's an exergonic reaction. And they're going to take that energy and they're going to couple it in an endergonic reaction to phosphorylate ADP to ATP. So they can do it just like we do in substrate level phosphorylation. Our mitochondria make the majority of our ATP by chemiosmosis. In fact, the whole process is called oxidative phosphorylation. We have an electron transport chain. We use high energy electrons to create a proton gradient. That stores chemical energy, like water behind a dam. Then that water flows through a dam. We use that to generate electricity. Inside the mitochondria, okay, across the inner membrane, protons flow through ATP synthase, and that makes ATP. That's chemiosmosis. Prokaryotes, instead of having mitochondria, they have an outer membrane, okay? They have a cellular membrane. Their electron transport chains and ATP synthase are on that outer membrane. They're on their cellular membrane, not in a membrane inside a mitochondria. Does that make sense? I hope so. So instead of having a mitochondria inside of them, they're just making it across. They're just using the electron transport chains and ATP synthesis embedded in their outside cellular membrane. That's it. That's why they're limited in size. They get bigger and bigger and bigger. They run out of ATP. Whereas in a eukaryotic cell, the almost every diagram you ever see of a eukaryotic cell isn't exactly right. They show like 4 or 5 mitochondria. The reality is you have thousands of mitochondria in a metabolically active cell. So that's why eukaryotic cells can get so large, because you can just keep packing in mitochondria. And in fact, about 10% of your body weight is in mitochondria. Okay. Okay. We got cofactors and coenzymes, Midnight Rider. Basically, you've got an enzyme, right? And these things facilitate chemical reactions, both endergonic and exergonic. And the classical physics model of this, or the model is it's got the active site that brings in the substrates, which are basically the reactants, and then lines them up to create a stable intermediate state. And then you have this input of energy that gets over the activation, which gets over these energy barriers, and then you form the new molecules with that. Okay, so you've got coenzymes and you've got cofactors. The reality is many of our enzymes really have a metal cofactor in there, okay, like a magnesium or an iron or something like that. And it's that metal ion that's actually facilitating a lot of that chemical reaction, we think, unless there's quantum tunneling. But that's what's facilitating that chemical reaction. And then a coenzyme is like an organic molecule that may not be one of the 4 macromolecules. There's a lot of things that aren't one of the 4 macromolecules. But a coenzyme basically helps give an assist to the enzyme. So think of like a good example of that would be coenzyme A, right? Coenzyme A takes acetaldehyde from the pyruvate oxidation and brings it into the Krebs cycle, the citric acid cycle. It adds it to this molecule called oxaloacetate, and the coenzyme A and then the citrase adds that acetaldehyde to it and forms citrate or citric acid. And so the coenzyme here would be the coenzyme A, and we call it acetyl coenzyme A because it's of course adding an acetyl group to it. So that's kind of like the difference there. Cofactors are like, these are metal ions typically at the heart of our enzymes, and then a coenzyme is another organic molecule that's assisting the enzyme there. I don't actually have a lot on that one, so yeah, I hope I answered that question for you. I didn't really focus too much on those. All right, you guys got some really good questions tonight. Does anybody else have any other questions they wish to ask? If not, I'll sign off, and I'll be on email a bit tomorrow, and probably I'll probably honestly be on email a little bit on the weekend. A little bit spotty. It's going to be pretty nasty this weekend, so I'm taking a little bit of tomorrow off, but we'll probably be working a little bit more on Sunday, so I'll be around. Okay. If you're building on number 7, if you're building a large molecule, explain how endergonic reactions can proceed with the enzyme with the help of a little bit, right? So if you have an end reaction, that means a couple things. You're taking these simple reactants, think photosynthesis, carbon dioxide and water, and you're going to make a more complex product like glucose. Okay, you need an enzyme to make this happen, because you could have water and carbon dioxide like around us all the time, and you can stay here to the end of time and you'll never get glucose precipitating out on your desk, right? Even though there's plenty of carbon dioxide and water in the room, you'll never get glucose molecules coming out of that. So you need an enzyme to facilitate this chemical reaction, and you also need a source of energy. And the way that our cells do this is they do what's called energy coupling. All right. So the energy coupling is they will couple an exergonic reaction with an endergonic reaction. That's also what happens in substrate level phosphorylation as well. When you phosphorylate ADP to ATP, the exergonic reaction is the oxidation of glucose. The endergonic reaction is the phosphorylation event. In the Krebs cycle, in the Calvin cycle, when you're reducing carbohydrates, this is an endergonic reaction. So what happens is a lot of times in these endergonic pathways, so a metabolic pathway is a series of chemical reactions. So like Krebs cycle, glycolysis, Calvin cycle, these are metabolic pathways. So in Calvin cycle, what happens is you'll take an intermediate and you'll phosphorylate it. And as I said earlier in this lecture, whenever you phosphorylate something, you add potential energy to it. By adding potential energy to a molecule, it makes the bonds easier to break. So like in the Calvin cycle, you take ribulose, which is a 5-carbon sugar, ribulose 1,5-bisphosphate. That's telling you it's got a phosphate on the carbon number 1 and a carbon number 5. So it's got a phosphate at each end of the molecule. And those phosphates are repelling each other and all the oxygens in there, which is giving that molecule a lot of potential energy. And that makes it easier for ribisco, ribulose bisphosphate, carboxylase, oxygenase, carboxyl, ACE telling you it's an enzyme. Carboxyl is telling you it's taking carbon dioxide and sticking it onto RUBP as a carboxyl group. And the way we get around that is we can have this activated intermediate, right? And that is caused by on that. So if you look at the slides, I go into a lot of detail about how that actually happens. And there's another good example in the book on making there's an amino acid. It's called glutamic acid. I think that's a precursor. But glutamine is like one of the most abundant amino acids in the body, and it shows that you take an ATP and the hydrolysis of ATP to ADP plus a phosphate group is an exergonic reaction, and then you take that phosphate, you create an activated intermediate, and then so the reaction of like actually adding the amine onto glutamate to make glutamic acid, I think that's right, goes from being endergonic to exergonic. So it's that energy coupling of these two reactions that really matter. Okay. Right. Is chemiosmosis phosphorylation down the electrical chemical gradient? I'm not sure what chemiosmosis phosphorylation. Okay, so chemiosmosis, chemi means chemical, osmosis really means to push because they're in osmosis. You know, water can push up the column. So what's happening here is, yes, the protons are flowing through ATP synthase down their electrochemical gradient. That is correct. It's like water flowing downhill through a dam. It has kinetic energy, and and the ATP synthase uses that kinetic energy to phosphorylate ADP to ATP. Okay, so I hope that answers it. Yeah, so the protons are definitely, in fact, as they push through the ATP synthase, that pushing through is they call that the proton motive force. I typically just call it the whole process chemiosmosis, but the protons pushing through is a proton motive force. Okay, really quick question. I've attended to be very bare bones. Like in more detail need to be. I don't want to lose points. You know, yeah, I mean, this is an open book, open note question, lots of time on it. You better not give a bare bones answer. Flesh out your answers well. And I do this not to like make it hard or anything, but there really is like an A answer, a B answer, and a C answer. You know, and a C answer is a bare bones answer. Really wrong. It's just incomplete. And you know, one of the reasons why I like details is because in our world, a lot of people repeat statements with no understanding of what that means. And I think it's important to have an in-depth understanding of a few key things in our life. And that way, when you realize like just how complicated something like why is plants green is, right? Then you start to realize, you start applying that to other aspects of your life that you may not have thought was complicated, but actually are, and you start to gain an appreciation for it. And second, it's a learning technique too. If you can learn something complicated here, get to somewhere else that's complicated, you know how to learn something hard, organize your thoughts, you can learn stuff that's complicated again. Okay. All right, I already went over, oh, got it, okay. I will definitely upload this to Learn. The lab I had a redox was a transfer of electrons. In the slides it says transfer of energy, which is correct, both. It's a transfer of electrons, a transfer of energy. So think about it. I'm taking glucose and I'm going to make ATP out of it, right? So glucose has potential energy, and you get 4 ATPs from substrate level phosphorylation. You get like 28 to 30 from chemiosmosis, and chemiosmosis is driven by the electron transport chain creating a proton gradient. The electron transport chain gets their electrons from NADH, right? And then NADH is carrying those electrons from the glucose molecule. So actually, the majority of your energy is carried by those electron carriers. And in fact, if you look at some of the videos I've got in the PowerPoints on photosynthesis, it's pretty wild. You realize that, I forget the math right off the bat, but a molecule of glucose has 586 kilocalories a mole. An ATP has 7.3. I think there's like 18 ATPs used to make a molecule of glucose in the Calvin cycle. That's only like I don't know, let's do the math. It's like 190, 170. Oh gosh, I'm gonna get myself in trouble here. Yeah, I'm not gonna try it. It's like under 200 kilocalories a mole. So there's like, you need another 400 kilocalories a mole. And you realize that there are these NAD pHs coming in to the Calvin cycle, and those were the electrons ripped off from water and elevated in energy. And you start to realize like the majority of the chemical energy stored in glucose came from this reduction of those high energy electrons carried by the NAD pH that were energized by sunlight. So yeah, so redox is definitely, I always like to think electrons, electricity, energy. The things go hand in hand. Yeah, this video will be made available in Learn. Yes, I will. It's available on my website too. I mean, on my YouTube channel, it'll be there, and I'll just post a link right into Learn for you guys. Okay. Anybody have any other questions here? Well, thanks for tuning in. This has been kind of nice doing some live chatting here with you guys. Good luck. You're welcome. I'm happy to help out. I'm happy to help out. Yep. Oh yeah, I'm definitely enjoying the new baby. The wife's in there with her right now. The other little kid and the 2 dogs. We have turtles too, and yeah, chickens. Man, I live on a real farm out here, way out here. And you guys are really welcome. Like I said, it's my pleasure, you know, to interact. Like I said, I just kind of wish we could have gotten to know each other a little bit in the classroom. But yeah, we'll do this, and then maybe we'll do another Zoom meeting before too long. My internet resets in 5 days, so I actually have a little bit more bandwidth, and I can have a little bit better video in about 5 days. And maybe we can and do another Zoom meeting again. A few people showed up the first time. Okay. Yeah, I'm happy to give you guys extended time on the exams. You can expect it for number 4 as well. That'll be fine. I can have it come out Tuesday or Wednesday. It was going to come out on Tuesday. Man, life can be hard out here sometimes. I got really sidetracked with some stuff. We had to go all the way into Albuquerque. Yeah, everything's fine. Okay, guys, I will. Good luck on the test. I'll be available a little bit on spotty over the weekend, but I will check emails at least once or twice over the weekend. And let's do a Zoom meeting at some point and get to see each other a little bit other than just you looking at me. Okay. Are there only 4 I can't speak anymore. 4 exams? Yes. And in fact, you can drop, this is the 3rd exam. You can drop the lowest one on 1 through 3. The 4th one is the final. That is it. I'll get it with Bruce. We'll get the exact format of that down, whether it's worth 75 points or 100 points. You can't drop the final, but it will be mostly over some review stuff and over chapters 11 and 12. So that'd be good. So the good news is, as we head into the holiday season, as we get tired with these classes, we kind of front-loaded this class a tad bit. So the next few weeks, there's actually going to be only 2 chapters to learn until now and the end of the semester. And that's nice because we got Thanksgiving coming up. Man, it's been rough out here this semester. So hopefully we can start to like pull back a little bit. And you guys are signing off. I'm going to sign off too. All right. Have a good one.